Good morning. So this morning we're going to have an interesting combined session. And it's one of the things that came up as a collaboration discussion last year. And we're trying to put forth together this concept of both highlighting some of the collaborations that we have going with ELGRA scientists, and also using the opportunity to combine again the physical sciences and the biological sciences, showing how we are all kind of looking at the same problems with different approaches. So first up, we're going to have Dr. Ricard González-Cinca, and he is the president of ELGRA right now, and he is also a fluid physicist. And so he's going to talk a little bit about some of the research in fluid physics that he has done, and also tie it together with our community just a little bit. All right, Dr. Cinca, please.
Good morning. Thank you, Annalisa, for the presentation and for the invitation to this nice conference. Yes, today I'm going to talk about two different topics. On the one hand, I will explain you the research that we carry out in the microgravity lab, in the UPC microgravity lab in Barcelona. But before that, I'm going to introduce you ELGRA, the European Low Gravity Research Association. It's going to be short, but if I wanted to make it really very short, I would say that ELGRA is the European ASGSR. But let me explain you a bit more about us. So ELGRA was born in July 1979. This is this year, last June, was our 40th birthday. So maybe I should now change and say that ASGSR is the American ELGRA, because I think you're a bit younger than us. ELGRA came from the idea of 7 European scientists, you have their names here, who considered that they wanted to form an association to foster the cooperation between European researchers interested in gravitational research. The kind of activities that we do, or the kind of association that we are, I think it's quite similar to ASGSR. We are a non-profit international society of multidisciplinary character. We're devoted to the promotion of European research in gravitational, in microgravity, in altered microgravity, or in hypergravity conditions. And in fact, we are a platform for all scientists who are interested in life science or in physical science in space. We aim to represent and strengthen the scientific community of gravitational research. At the same time, we want to involve young people in space research by participating in educational programs, in our educational programs, or in European Space Agency educational programs. We have a similar type of members as you have in ASGSR. Our members are individuals who are engaged in scientific research, in gravitational research, or scientific institutions or companies already also interested in gravitational research. We act organizing scientific symposium and courses. We organize a biannual conference. The last one took place last September. Next one will be in fall 2021. We cooperate with institutions and agencies in the organization of scientific events. We spread the information about any kind of announcement of opportunities that we know about, and new platforms, space policy to our members, etc. And we support the students and young researchers to attend scientific meetings by means of grants. And we also collaborate with the ESA educational programs, Fly Your Thesis, Spin Your Thesis, Drop Your Thesis. Every time we meet in the biannual symposium, we award medals to outstanding scientists in both life science and physical science. We also have a gravity science image gallery, very similar to the one you have here in the lobby, where we ask for our members or participants in our symposium to show the nice pictures. After the symposium, selected papers presented in the symposium are published in the journal Microgravity Science and Technology. This is not a journal of our association, this is a Springer journal, but every time they issue, they prepare a special issue on the papers presented in the symposium. Every 2 years, we present a summary of our activities in what used to be our newsletter, which became recently the magazine with a more modern format. And we also have a student association, ELGRA, that was created only 2 years ago, but it's a very active association, and they are organized many activities for the students, and they provide travel grants to attend meetings, etc. Finally, I would like to mention a hot topic. I would say it's something that will finish today, which is the following. Next week, some of you may know that there is the European Space Agency Ministerial Council. This is the meeting where European Ministers of Science decide the funding for the next 3 years in European Space Agency. So it's a very important meeting. So we prepared a statement at ELGRA, which has been signed by many scientists, in order to foster or to support the program presented by the European Space Agency. So maybe at the end, if I have time, I can show it. I can show the link again in case you want to sign it. So now I completely changed the topic, and I'm going to talk to you about the research we carry out in our lab. Actually, I wrote a subtitle which is From Bubble Generation to the Removal of Bubbles. Why? We are interested in two-phase flow management. We carry out both numerical and experimental studies on systems in two-phase flows in different gravity environments, from microgravity to hypergravity. Our initial interest was focused in systems like bioreactors, but also in fuel tanks. We wanted to learn more about the injection of bubble jets and the injection of liquid jets. To this end, we characterized a bubble generator, and we studied the injection of these bubbles. We analyzed the flow pattern generated in the capillary of the injector, and in general, the dynamics of bubbles, bouncing, coalescence, etc. Later, as you will see in this presentation, we kept our interest in this type of systems. This is bioreactors or fuel tanks, but our aim was slightly different. We didn't want to inject bubbles, but we want to remove them. Or we want, in the case, that was in the case of the fuel tanks, what we wanted is to manage boil-off, which means to be able to detach bubbles from the hot spots in the fuel tanks and to bring them to cold regions where they could collapse. As regards to the life science application, we would like to do, and this is what has been doing for the last times, we wanted to be able to degas solutions. This is to extract, for example, oxygen in a biological solution. So our studies started, as I said, with the consideration of a bubble injector. In fact, this is a T-junction. You have two versions. One version, you inject liquid from the top capillary and gas from the top and liquid from the horizontal capillary. And you also have the inverted version in which you inject liquid from the top and gas from the horizontal capillary. What happens here is that the liquid drag breaks the bubble, breaks the air bubble, squeezes it and breaks it, so that you manage to generate a two-phase flow in the capillary behind it. What we did for the characterization of this injector was to study the role played by the liquid and gas flow rates. Okay? And we saw that these were the two main parameters to determine almost everything in the two-phase flow generated after, which was, for example, the frequency of generation of bubbles. We are able to control this frequency of generator just by playing with the flow rates, and also the size of these bubbles. We are able to determine the size by fixing the flow rates. On top of that, we managed in these bubble injectors to obtain different flow patterns, from bubble flow to slag flow or charm flow. We carry out some, we did a numerical model, a volume of fluid model to, sorry, let me see how can I, sorry, trying to play this. Yes, so here you see the generation of a bubbly flow in this T-junction. Sorry, this is, yes, here you have the picture of the squeezing of the bubble and the breakup of this bubble experimentally and numerically. And at the bottom, you have both experimental and numerical results for the generation of a bubbly flow, generation of a slack flow, and the generation of a charm flow. So once we knew how to generate these two-phase flows, we wanted to see how will they become if we inject them in a tank full of fluid. For example, these are some examples. When you inject this two-phase flow in the tank, the structure of the bubbly flow that you will have in the tank will depend on the two-phase flow that you had in the capillary and also on the characteristics of the nozzle that is injecting this two-phase flow. As you can see here, these are on-ground pictures, but you have really different type of structures. Not only that, if you are using two injectors, two nozzles, and this structure will also depend on the orientation. Here, these two nozzles are oriented at a certain angle, while here these two nozzles are almost parallel. So the orientation of the nozzles, the distance between the nozzles will also determine the structure of the two-phase flow inside your tank. We also perform some numerical simulations in order to reach a larger range of parameters, both on the ground and in microgravity conditions. Since we already had an experimental setup to deal with different nozzles, we thought that could be interesting to analyze also the injection of droplet, the liquid injection. So this is what we did afterwards, which was both on the ground and in microgravity conditions. We observed the liquid injection, and in fact, we distinguished between the dripping regime and the jetting regime in microgravity. We also got interesting images that you can see here. When you use two nozzles, you get interesting patterns depending on the orientation of these nozzles, whether you can see these nozzles, these jets impinging in a weird pattern, or you have here a big droplet, a kind of a liquid bridge with a different orientation of the nozzles. Coming back to the bubbles, we were also interested in studying the rise of the bubble and the collision with a free surface. The collision of a bubble with a free surface is kind of a coalescence phenomenon. So we studied the bubble rise at different gravity levels, from 1G to 18 or 19 Gs in the larger diameter centrifuge at ESA. And we observed that the trajectories of the bubbles, as you can see here, very much depend on the gravity level, as you can also observe in these movies. On the left, sorry, don't manage to. Okay, so you see both here. On the left, it's at 2 Gs, and on the right, the bubbles rise at 15 Gs. The bubble rise at 2 Gs is a rectilinear rise, while the bubble rise, or almost rectilinear, while the bubble rise at 15 Gs, it shows a zigzag trajectory. After this rise, the bubble impacts with a free surface. This impact can have two different types of consequences. Either the bubble coalesces with a free surface, or the bubble can bounce with a free surface. In fact, this depends on the bubble size. The smaller the bubble size, the more likely to immediately coalesce with a free surface. for large bubbles, what we see is a bouncing 2, 3, 4 different bounces, as you can see here in this boundary element method simulation, where we saw the bubble rise and also the bouncing of the bubble with a free surface. We were not able to simulate coalescence because this is a microscopic phenomenon which is not contained in this boundary element method. So after this knowledge we acquired on the generation of bubbles, on the behavior of bubbles in different types of systems, we decided that it could be interesting to see what happens if we change pressure in our system. How can we change the pressure in a bubbly tank, in a tank with liquid and bubbles? So what we can do is to use an acoustic waves. So the aim was to try to manage two types of systems in principle different, which are the boil-off or the management of boil-off in cryogenic propellant tanks and the electronic cooling. These are systems with at some point there's some boiling generation and some bubbles that in principle they remain attached to the hot spots, and something that in principle you don't like. You want to manage this, both from the point of view of mass transfer and from the point of view of heat transfer. So let me show you only a couple of slides on the theoretical background to understand this better. If you have a bubble in a liquid tank and you apply an acoustic waves, the consequence to the bubble will be in two different ways. On the one hand, the bubble will oscillate due to this pressure change. It will be an oscillation of the bubble volume. On the other hand, since you have a gradient of pressure inside your tank, there will be a translation of motion of the bubble inside the tank. The combination of these two effects are contained in the primary Bicharnes force. This force tells you which are the acoustic effects on your bubble. The fact that the bubble is oscillating when it's under an acoustic field makes that in turn this bubble will generate a secondary acoustic field, of course much weaker than the primary acoustic field, but an acoustic field that will have to be considered in case there are neighboring bubbles to this initial bubble. So the secondary acoustic field generates a secondary acoustic force to these neighboring bubbles. We can consider two types of acoustic waves to control these bubbles. We can talk about traveling waves, or we can talk about standing waves. Basically, what do a traveling waves to a bubble? The traveling waves, what will do will be to push the bubble away from the generator of the acoustic waves. It will be pushed away. While the standing waves effect will be slightly different. You will have some nodes and antinodes of the acoustic waves, so that the bubble will have a different behavior depending on its size, or depending on the resonant frequency of the bubble. The resonant frequency is determined by the size of the bubble, by the radius of the bubble. So what happens is the following: is the frequency of the acoustic waves is smaller than the resonance frequency of the bubbles will be driven towards the antinodes of the acoustic waves, toward the positions of the antinodes of the acoustic waves. If the rest of the frequency of the acoustic waves is larger than the resonance frequency of the bubble will be moved towards the nodes of the acoustic waves. But in any case, it will be moved to a certain position, and once the bubble will reach this position, either the node or the antinode, it will remain fixed there, because the acoustic force there is 0. Okay? So it will levitate there. These effects in bubbles, the acoustic effects in bubbles, can be seen at any gravity level, provided that you use a sufficiently large acoustic amplitude. Here on the left video, you can see here bubbles rising at 2Gs without an acoustic waves. And here are bubbles rising at 2Gs with a vertical acoustic wave. Okay, so it's clear that the trajectory is influenced by these acoustic waves. How it is influenced, we can discuss it more in detail in another moment, but it's influenced, that's clear. But not only at 2Gs, but at 19 Gs. We apply an acoustic wave of sufficient amplitude, and you can see on the left, bubbles rising at 19 Gs with acoustic waves, and on the right, you see that the trajectory is totally different. What is the influence of these acoustic waves in the bubble behavior? We can say that the injection direction can be changed. Also, these oscillator trajectories are influenced by the acoustic waves. We see a deviation. We can see bubble levitation. At a certain point, the bubble can be trapped at the nodes or antinodes. We can see levitation if the amplitude is too large, which at some point it cannot be good. And we, of course, see this secondary Bichner's force effect, this bubble-bubble interaction. So if we want to try to simulate this behavior numerically, we will have to build a model which will contain, on the one hand, the translational motion of the bubble, and on the other hand, the bubble oscillation, the volume oscillation of the bubble. So in the translational motion of the bubble is represented in this equation where you take into account all the forces acting into the bubble, with or without gravity. You can turn it in or turn it off. And the bubble oscillation is represented in the Rayleigh-Pleasant equation. So we perform some numerical simulations in a system in which the acoustic waves is parallel to the gravity force, and we could turn it on and off, as I said. And the first observation is this one. You have in red the position of the bubble at 0G, and in blue the position of the bubble at 1G at different times, okay, as a function of time. So you see that there's an equilibrium position of the bubble in both cases. This means that this position at 0G is exactly the node of the acoustic waves, while this position at 1G is the place where the buoyancy force and the acoustic force are balanced, which is not going to be exactly at the same position as the node. But in any case, what we can see here is that the acoustic amplitude was large enough to be able to compensate the buoyancy and make the bubble levitate somewhere, not in the node, but somewhere else. We did different simulations to see the effects of pressure amplitude, and you see here, for example, that at low pressure amplitudes, at high pressure amplitudes, bubble will stabilize, will levitate at the node or very close to the node, just a bit above the node. While if we go to high pressure amplitudes, low pressure amplitudes, the bubble, the acoustic force will be not sufficient to trap the bubble, and the bubble will keep rising thanks to the buoyancy force. Here, basically, what we have is images at different injection points. The bubble size does also play a role, of course, as you can imagine, because the larger the bubble size, the larger the buoyancy force, therefore the less acoustic effects on the bubble. So large bubble will tend to rise, while small bubbles will tend to be trapped by the acoustic force. Now, here I will show you an experiment carried out in a suborbital flight. A suborbital flight, by the way, with a very high G jitter, which disturbed very much our experiment, but we still could make some observations. The idea here was to use acoustic waves of a changing frequency. So what we wanted to do was to move bubbles from one node to another node. So we started nucleating bubbles, and with a frequency of the acoustic waves as 168 kilohertz. So bubble were close, the node was close to the wall, to the hotspot. Then we changed the frequency, and you can observe these red and blue bubbles that move towards the node at 35 kilohertz. And they stay there. There are some oscillation, as I said, there was a strong G jitter. Later, what we did was we changed to 162 kilohertz, but a lower amplitude, and that was not enough to compensate the G jitter, and the bubble trajectory was driven by the G jitter. But at least here, what we managed to show was that we can move bubbles wherever we want, provided that we use the right frequency, therefore the right node position. Finally, in this last video, what we did was to apply an acoustic wave, but in another direction. Instead of applying the acoustic wave perpendicular to the heater, we applied the acoustic wave parallel to the heater. So what you will see here, this is a drop tower experiment. You will see bubbles being injected by a 1 millimeter heater. And of course, at the start, they will rise. And once we drop the capsule and with the acoustic waves on, you will see that bubbles are trapped at a certain position, which is the position of the node of the acoustic waves. So you see they are rising, and at some point you will see that they are trapped. Now the capsule is falling, and bubbles are trapped at the nodes of the acoustic waves. So the idea of all of this is to be able at some point, or to mature a technology that makes us able to control nuclei boiling in long-term cryogenic propellant storage. I'm talking about, for example, fuel depots, or I'm talking at cryogenic fuel tanks. So this is a movie that we made in our lab, where the idea is, I think it's very clear. So you have this fuel depot. There will be some hot spots somewhere, which will create some bubbles, and where we will apply an acoustic wave, which will allow us to detach these bubbles, to move them to the nodes or antinodes, depending on their size. And if these nodes and antinodes are in regions sufficiently cold and the bubbles are sufficiently small, these bubbles could collapse. They could condensate. So they will recover them as liquid phase, in liquid phase. So this is all. I would like to acknowledge my PhD students who have been working in these projects, as well as the collaborators and Spanish Ministry of Science, ESA, French Space Agency, and NASA Fly Opportunities Program. Thank you. I can hear you.
Thank you very much. I'm not a physicist. I'm a biologist. So this is really an eye-opening chance for me to sit and think about the impact of this. And I'm going to piggyback off of what Annalisa said in her introduction about the importance of crossover between physics and biology. 2 weeks ago, there was a publication by some people who were measuring blood flow in a pipe, the jugular vein in astronauts, and found that it was profoundly affected by spaceflight. I'm wondering if there's anyone in your community, or if you know of anyone in our community who are looking at this in a more applied way to understand the flow of blood through pipes, which are not static, but changing in frequency and and dynamic. And I'm wondering whether the model can be applied in that way. And I'm sitting there thinking, we don't have bubbles in our bloodstream, but we do have.
But we could have them. Well, we don't want to have them. But
well, it's a very complicated situation, and I don't know how much physics can. Physics obviously can deal with it and help us understand it. There are there are cells, there are molecules of cholesterol and whatever else. So, I mean, it's a very complex situation. Are we equipped with the tools to be able to model and predict those changes? And I don't know, for those of you who saw the article, or even in the popular press, it was, blood flow in the jugular vein in astronauts was flowing backwards.
I know there are some research teams. I think they are in France. They are biophysics teams, so there are some of the researchers are physicists, others are biologists, which is ideal. So I know that sometimes they have problems in the vocabulary they use, but they know they are working on the blood flow in human body. I don't know if they have studied this particular problem that was in the press last week, but I'm sure that they could be. And I would say that from the experimental and especially numerical point of view, it's much easier to deal with single phase flows than with two phase flows. So in principle, it should be feasible.
Well, and I'm sure I'm not the only one who thought of this, but you know, we spend a lot of effort and collaboration with physical scientists in studying the flow of water to water plants. But I don't think we think as much about what's going on inside the plants, because fluids have to move from one part of the plant to the other, and the impact of being in microgravity must have an effect. Maybe I'm just slow, but I hadn't really thought about it in that way. So thank you for the stimulating talk.
Thank you. I would say that I don't know about plants, but perhaps an advantage is that maybe we could consider almost isothermal systems. I don't know, which is an advantage compared to some of the systems we are dealing with. So if there's no heat transfer, or very little heat transfer that you can neglect, that's a huge advantage. Thank you.
We're gonna have to move on. So could we please have questions maybe at the end or at the break? Thanks for understanding. All right, next up, we're going to have another member of ELGRA is Dr. Javier Medina, and he is the head of a laboratory in biological sciences associated in the Spanish National Research Council, and his research is focused on plant research, and in fact, his research was just recently recognized with the ELGRA medal for his innovative research in microgravity on the ISS.
Thanks very much, Annalisa, and good morning to everybody. First of all, let me express my gratitude to the executive committees of both the American and the European societies for this exciting initiative of exchange of ideas and exchange of results. Really, I think this session expresses a thing that already exists, which is the collaboration across the Atlantic Ocean between researchers of one or the other side of the sea. But it is good, I think, that this kind of collaboration is not only visualized, but also strengthened. And well, for that reason, we are here. And on my case, I will show you some of the results obtained by my group in Madrid in collaboration to many other teams and laboratories, both in Europe and also in America. So, well, the key question is for us, for plant biology researchers, our job is to deal with plants in space. And the question is why we do this. Why should we bring plant to space? Probably most of you know the solution, but I could summarize this answer in two key objectives. First, the obvious, the deal with many times throughout the conference, which is the contribute to make possible the human exploration of space, but also we should not miss the second objective, which is related to the basic science, to the basic biology, which is to learn essential concepts on plant physiology and stress response. A few key questions that emerge from these two objectives are, for example, what are the alterations caused by the space environment on plant growth and development, whether or not plants are able to survive and to live and to reproduce and to extend their lives in an environment so different to the Earth environment as is the space one, if it is possible or not to optimize, to do something in favor of the adaptation of plants to the space environment, and also, last but not least, we live in the Earth, we use plants in the agriculture. So the last question is, the last essential question is if we can use this knowledge to improve the culture of plants on Earth. We have, throughout the conference, this conference, we have been able to know about many different studies focused on very different processes and mechanisms that occurs in plants as an organisms, as plant cells, and so on. Our particular focus is put in the root meristem. The root meristem is, well, is a part of the root tip, a collection of cells, and these kind of cells are specialized in providing, in continuously dividing and growing. They are involved in the cell cycle. The function of the root meristem is essential for the plant root, for the plant growth and development, because they are the provider, the suppliers of cells for the differentiation, which is the essential process that occurs throughout the plant development. This occurs because these kind of cells, meristematic cells, are continuously engaged in the cell cycle, growth and division, and in a certain moment, one of these cells escapes from these cell cycles and enlarges and endoreplicates the DNA and finally gets the differentiation. This occurs throughout all the entire life of the plant. So this kind of meristematic cells, the pattern of differentiation and the pattern of development is mostly fixed and mostly depending on the situation on this essential tissue, the root meristem. There is an important relationship between the gravitational perception, the gravitational transduction of the gravitational signals and the status of the root meristematic cells. In fact, we know that the gravitropic signals are perceived and they are transduced to the meristematic population of cells throughout the balance of the phytohormone auxin. Auxin is an important transducer at the level of the full organism of the plant. It's an important transducer of the gravitational signal, but also is an important driver of the meristematic function, in such a way that throughout the activity of some intermediates, auxin ultimately controls the so-called meristematic competence. Meristematic competence is the balance between the process of growth and the process of division. So if meristematic cells are really competent, they can do their function. If they are not, the function of the meristematic tissue and finally, ultimately, the plant growth and development as an organism, at the level of the full organism is seriously compromised. Our first experience with the influence of the space environment on the function of meristematic cells was taken from the so-called root experiment. The root experiment was performed some years ago in the so-called Cervantes mission, was one of the Soyuz missions that were promoted by different European countries at the time in which the International Space Station was still in the process of assembly and the first operation. And in fact, the root experiment was the first European experiment carried out on plant biology. In this, essentially, this experiment was very important because really we got from this experiment the settlement, the first important ideas on which we elaborated further with different approaches, with different complementary approaches. And this basic knowledge that we approached, that we got from this experiment was that meristematic competence was disrupted by microgravity, was disrupted in the space environment, in such a way that using at that time only microscopical techniques, only the cellular approaches to the study of this process, we got the conclusion that cell proliferation is enhanced in microgravity, whereas cell growth studied by means or marked by means of the ribosome biogenesis, the process conducing, allowing the protein biosynthesis, cell growth is depleted. So these two essential processes, which are the basis of the meristematic competence, were disrupted. This initial result was complemented and confirmed, and we got a lot of additional knowledge about it by using the so-called ground-based facilities. We have used, for example, clinostats magnetic levitation, the random positioning machine, and also we not dismissed the effects of the opposite gravitational situation to that we found in space, which is hypergravity by means of the use of the large diameter centrifuge. These kind of facilities are capable of providing flexibility and a good level of reliability regarding the effects of the true microgravity that we got in space. Together with these results that we got on incubating seedlings in this kind of facilities, we did a step forward by means of the use of the plant cultures. The use of seedlings really has some, well, in fact, the use of seedlings or the use of plant cell culture has advantages and disadvantages. But really, we took advantage of the use of this kind of system because we were trying to test whether or not this response of the meristematic cells, of the dividing cells in general, non-differentiated cells, to the microgravity environment was really a question of the general management of the function of the whole plant, or was purely a cellular function. And this is important, well, mainly in the context of this conference, because the question on whether or not, well, the well-established systems of gravity perception by plants based on the statolith function at the root tip was the only system of gravity perception or not. Really, the idea that there are additional systems at the cellular, based on cellular functions, complementary to this system of gravity perception, is really emerging, and is practically, well, considering the results that are being published now, really practically nobody doubts in that really together with the system of statolith function there is another system at the level of cells which is responsible for an additional perception of gravity. This is very interestingly, the use of cell culture is very interesting for that function because we got from this a pure cellular response to gravity alteration. Otherwise, the use of this kind of systems provide us with lots of homogeneous populations of cells, which are very useful for the use of molecular biology techniques. Well, we took this kind of cell cultures, and the first thing we did with them was to synchronize in the progression throughout the cell cycle. We use for that the drug called hafidiccolin. The drug blocks the cell cycle at the level of the entry into the S phase. The S phase is the duplication of DNA phase. And once the drug is removed, the population progresses synchronically throughout the cell cycle, so that we can get samples at different times. We are sure that after a flow cytometry control and flow cytometry analysis, we are sure that we are getting exactly a certain phase of the cell cycle, depending on the time at which we get the samples. So once this system was well established, so we took the system and incubated in simulated microgravity by using the random positioning machine. And for the analysis, we use flow cytometry, and also we use transcriptomics and microscopical techniques. Well, the first results that were very clearly shown in our study was that the cell cycle under simulated microgravity conditions is decelerated, is shorter. In the cell cultures under simulated microgravity conditions is shorter. This was shown very, very, very clearly by means of the study of the inflow with the flow cytometry technique, in which we observed that the entry in mitosis, the G2 phase, was accelerated, was shortened under these conditions, was strongly shortened, whereas the G1 period, the period which occurs after mitosis, was slightly enlarged, slightly increased. So the general result was that the cell under simulated microgravity condition, cell cycle is shorter. This is absolutely in agreement with the results that we obtained in the in the space experiments complemented with the simulated microgravity studies on seedlings. So the result of the disruption of the meristematic competence, at least related to the increasing of cell proliferation rate, was absolutely confirmed with this study. Not only confirmed, but also we entered into the reasons, into the mechanistic alteration. So we can say after this study that really the cell proliferation rate is enhanced under simulated microgravity conditions because the regulation of the cell cycle is seriously altered at the level of the entry in mitosis. So the acceleration of the cell cycle. Not only that, but also the cell growth was observed to be depleted. The study of the cell growth was done by means of the use of the ribosome biogenesis process and using two essential markers of this process, which are two essential proteins called nucleolin and fibrillarin. There are two key regulators of the ribosome biogenesis. We performed a study at the level of the gene expression and also at the level of the protein level using flow cytometry for this purpose. And in both cases, we observed, depending on the phases, the situation exactly of the gene expression and the protein level of these two important regulators of the cell cycle in the different phases. And the result was that the ribosome biogenesis was depleted under simulated microgravity conditions. So really, the data that meristematic competence is disrupted under altered gravity conditions was absolutely confirmed by means of this study because cell proliferation was increased due to the shortening of the cell cycle and cell growth was depleted due to the alteration in the gene expression and the protein level of these two essential regulators of the process. You can see here a schematic view of the regulation of the cell cycle. We did a transcriptomic study, using the so-called core cell cycle regulators. We got a full list of regulators at the different checkpoints of the cell cycle, and we observed the status of the gene expression, of the transcription of these genes at the level of these checkpoints of the cell cycle. And the results clearly expressed was a upregulation of the regulators of the genes that perform the checkpoint at the G2 point, and down regulation of the genes that act at the level of the G1S transition. So this is confirmed the alteration of the cell cycle regulation as the essential cause, as the essential factor producing the disruption of the meristematic competence. Not only that, but also the general level of transcription of the meristematic cells was shown to be depleted under simulated microgravity conditions, and this which was got by means of a study with using RNA polymerase 2 immunocytochemistry. RNA polymerase 2 is the enzyme responsible for transcription of the extranuclear genes, so it's the enzyme responsible in general for the transcription of the genome. And the study in situ showed a depletion of the level of transcription by this enzyme, accompanied by an increase of the chromatin condensation. Chromatin condensation is associated to an inactive state of the chromatin regarding transcription. So transcription as a process was depleted and chromatin condensation was increased. Chromatin condensation is also associated with the so-called epigenetic regulation of gene expression. And we study also the situation under simulated microgravity in these systems of two essential factors producing the epigenetic regulation. Let's say the histone acetylation and DNA methylation. And we show that epigenetic regulation of the gene expression is also altered under simulated microgravity conditions. Well, the conclusion of all of this line of research is that microgravity, either real or simulated, produces a very serious alteration on the status of the meristematic cells. Taking into account that, as I told to you at the beginning of the talk, that the status of this kind of cell is essential for the pattern of plant growth and development, the conclusion is that really the microgravity conditions alter seriously the life of the plant. So compromise seriously the plant development if these alterations that we observed at the level of the meristematic tissue are extended throughout the life of the plant. So really this should seriously compromise the viability of the plants in these conditions. However, complementary studies, not performed by our group, but the experience of the plant biology community, showed that plant did survive in space, and did survive not only until the adult stage, but also until, for example, the reproduction, as observed with the growing of these nice flowers that, from the point of view of the common population, are very beautiful elements, and from the point of view of the biologists, are the reproductive organ of the plant. So plant really survive. What's the question then? The question is how did they overcome the problems? So if plants did survive, this means that the plants have overcome the problems that produce the disruption of these essential processes. So we thought in that maybe there would be other factors that could replace the absence of gravity and producing overcome the adaptation of plants, which is finally the goal of the process. Other factors, other environmental factors could could help the adaptation. And we thought in the light at one factor in that question, because phototropism could help or could replace the absence of gravitropism in these conditions. You can see here the different possibilities of combining light and gravity as a factor driving the plant development. So this is an old slide that I used to show long time ago, because it was really the starting point for our next step in this career, was the knowledge that particularly red light, not only light, but particularly red light, is an important factor improving, promoting specifically the processes in that we are interested. So red light irradiation, we know that it enhances cell proliferation, it increases phosphorylation of nuclear proteins, which is a process, a molecular process associated to the process of transcription and cell growth. And red light in particular induces expression of nucleolin and cell cycle genes. This is an old result, but it is still important to know, because it is the starting point for interesting results that we have got later. In this context, we have performed the seedling growth project, a complex international project in the International Space Station that interestingly was a joint venture taken by both agencies across the ocean, NASA and ESA. Seedling growth had an American PI, Professor John Keyes, and an European PI, which is myself. This was the working hypothesis for that project. As you can see, it is an elaboration of the previous schematic view that I showed you before about the relationship between gravitational signal and the meristematic competence. We have introduced, well, the former schematic view was this line, using the so-called professional cells. So that means cells specialized in the gravity sensing, but also in the plant there are non-professional cells, cells that apparently don't have systems for gravity perception, but they have. And also how red light irradiation could interfere with this mechanism and finally produce a restoration of the meristematic competence. Seedling growth consisted of 3 parts that we have, you can see here. Was performed throughout some years between 15 and 18. Was performed in the Columbus module in the ISS using the European Modular Cultivation System and the Tropic hardware. There were 2 sets of samples and 2 procedures of growing seedlings. One of them was proposed by the American team and used this scheme ceiling growth using a collection of genotypes associated with light perception, mostly consisting of phytochrome mutants. And in our case, we propose the use of this scheme of ceiling growth using a collection of mutants, of genotypes, especially affecting plant cells and proliferation, so finally arriving to this kind of conditions of using plants. In addition, we use a new device produced by a Spanish industry to make possible the fixation of plants, of ceilings in a space which was called fixed box. You can see here the growth of seedlings belonging to the second part of the collection. The first image was taken at the end of the photoperiod white light illumination, and you can see here how under 0G and 1G seedlings are developed in the presence of microgravity conditions, in that case, real microgravity, and the 1G condition in space provided by the centrifuge of the EMCS in the other part. You can see, well, clearly the different ways of developing the seedlings throughout these 2 last days in which the seedlings were illuminated with red light. After the G1, the seedling growth 1 experiment, we performed some analysis with using qPCR on specific markers of the oxygen transport, of the cell cycle regulation, and of the ribosome biogenesis process. And we got the result that specifically in the wild type and in the 5B mutant, the expression of all these genes was concerted. So under our previous studies, we have seen that this kind of genes, those regulating the cell cycle and those regulating the ribosome biogenesis, were disrupted, were expressed in different directions. In that case, we observed a concerted expression of these genes. In the seedling growth 2, we did a global transcriptomic study that was described by my colleague Raúl Herranz in a different session of this conference. But I would like to emphasize regarding this global transcriptomic study that we try to solve 2 essential questions. Is red light capable of reverting the pattern of gene expression under microgravity to a pattern close to that of control 1G conditions? And also, is red light irradiation capable of counteracting gravitational stress? So we use the GDI tool for that. GDI tool, I will not enter into the description of that, but gives a general idea of the gene expression level at the level of the full genome. We can see here how using the wild type under red light 1G conditions in space, so probably the optimal conditions of growth, this is the pattern that we observed. Under dark microgravity condition, the pattern is clearly changed, and under red light microgravity condition, the pattern is restored to the initial condition. So red light is capable of restoring the level of gene expression that we observed under controlled physiological conditions. Comparing between conditions, we get also a similar pattern when we compare the dark micro G between red 1G, so the extreme condition, the conditions of microgravity compared to the conditions of the red light illuminations under microgravity. We get similar results. And also, we took advantage of the use of nucleolin mutant, specifically of the NUC2 mutant, which is a sensor of stress. And using this mutant, we could get the result that the comparison between in the wild type with this mutant under red 1G condition is no stress. Nucleolin 2 does not detect any differences, but the situation in the microgravity is a high stress, and the situation of the microgravity with red light illumination restores the situation of no stress. Finally, we are in process of getting results of the ceiling growth 3, and in that part we can get how the oxygen transport is altered under microgravity condition. This is the control condition, and we also got the result at the level of the mass gravity. Under mass gravity, by the way, an interesting result that we have obtained, which is the last one that I will show you, is that under mass gravity condition, the orientation of the ceilings is done practically at the same level as it is done under 1G condition. So, and that's the result that I would like to show to you. This is my team in Madrid. This is a meeting on the scientific team of ceiling growth that took place in Granada a couple of months ago. We are very grateful to NASA for having awarded us with the Group Achievement Award. We are also grateful to our colleagues, our European colleagues at ELGRA, which gave us this award. You can see here the list of persons by using the NASA award that I express my deep gratitude, of course, but you can see here the list of people that, many people that took a relevant place, a relevant position in making this project to go, and finally, the gratitude to ESA, to NASA, and the Spanish Ministry of Science for having supported financially our activity. So thank you very much for your attention.
We're going to hold questions until afterwards, and we're going to move on to our next speaker. So now we start into our next section, but first, thank you very much.
Okay.
So I'd like to introduce our next speaker, Dr. Greg Loraine from Dynaflow Incorporated, who's going to talk a little bit about how integrating the systems that we use. All right, thank you very much.
Hello, thank you for joining me on the last morning of the conference. Today I'm going to talk about a piece of hardware used for separating gas from liquids, the DYNASWIRL phase separator. I'd like to acknowledge my associates, Chao Sung, Jingseng Ma, and George Shaheen. And of course, John McQuillan, our NASA technical advisor. So as we've heard throughout the conference, two-phase flow is an important aspect of space flight, as gases and liquids need to be recycled and reused. And the longer the duration of the flight, of course, the more important that becomes. Our approach to this is the DYNASWIRL phase separator. It consists of concentric cylinders. The bubbly flow comes in at the top and is injected tangentially through slots so that it induces a spin. Now, this can create a gas core at the center. So the bubbly flow will come in at the top and spin around. The liquid is exited out the bottom. The gas is extracted through, axially, through a gas permeable membrane. So the gas will go out the top and the liquid will go out the bottom. Now we have tested this in microgravity flights in 2009, 2012, 2013, and twice in 2015. So the system consists of a pump, the flow control. In the movie, you can see the bubbly flow coming in at the top, spinning around and being captured by the core. The air injection is at the upper right-hand corner of the still figure, or upper left-hand corner, goes into the swirl chamber, which is in the illuminated in green, and then returns through the system. So our first approach was to take numerical simulation. So this is 3D quarter domain. These are the non-slip conditions. We used a Navier-Stokes equation. The software we used was the DynaFS-Viz. We used the artificial compressibility. So this is an example of that simulation taken in right here at the middle. Now, the solid lines are the simulation results, and the symbols are from PIV made in the actual system. So once we convinced ourselves that we had good agreement between the simulation and the data, we then proceeded. One of the first things we looked at was the effect of the orifice diameter. 0.125, 0.25, 0.375, and 0.5 inch. So on the left are plots of the different orifice sizes. 0.5, the 3/8, the 1/4 inch, and the 1/8 of an inch. The figure on the right shows the maximal tangential velocity for each of those conditions, 0.5, 0.37, 0.2, and 0.125. So the smaller ones showed the highest tangential velocity. Now, the same simulations also gave us information on the pressure in the chamber. 0.5, 0.375, this is the 1/4, and this is the 1.25. So essentially the exit orifice was too small and created a back pressure in the system. So on the right is that pressure in the chamber. You can see it's much higher for the 0.25 or 0.125. So based on the pressure and the tangential velocity, we settled on the 0.25 orifice diameter. Our next question was, what would be the optimal diameter for these systems? Starting with 4.75 inches, this was our 1D. We took half of that and twice of that and ran simulations for that. And conserving everything was done with a 0.25 inch orifice. The lengths are the same. The inlet velocity or inlet slots are also the same. So this figure shows a simulation of the biggest chamber, the 1D chamber, and the 0.5 chamber. Again, this is tangential velocity. This figure is a close-up of this area. We can see that the 2 has a lower tangential velocity than the other 2, and the 0.5 was higher than the 1. Now, again, this is pressure within the chamber. This is for the 2 times, and the 1 and the half size are about the same. Again, a close-up of this region right here is on the right. We can see that the predicted lowest pressures in this vapor core, the lowest is with the 2D, and then the 0.5, and then the 1. So we then made test cells for this with the diameters of 2.5, 4.7, and 9. Again, the orifice sizes are the same, the injections are the same, tangential slots, everything else is the same, including the length. We then tested this system, selecting the liquid flow rates and then increasing the air addition rates at each flow rate. And we held the pressure constant for these tests. And we measured the separation efficiency by measuring the amount of air we added and the amount of air that was extracted. This criteria was the amount of removal, and also would it maintain that pressure anytime if the core failed, then we considered that test as a failure, or if the pressure increased beyond the set pressure. So this is for the 1D setup. We did 20 PSI, 30 PSI, and 45 PSI. So along the x-axis is the water flow rate in the system. The y-axis is the air flow rate. So the blue is 100% removal. The green to yellow is transition between total removal and partial removal. And the red areas are where the system failed, either due to core instability or at the higher flow rates, the addition of more gas exceeded this pressure. Back up. Again, this is for half size. We see the same kind of pressure with each condition. There was an optimal pressure with the gas addition. And this is for the 2D, again, same pattern. Now in this slide, we took the maximum removal efficiency at each flow rate and plotted them for each one. The 20 PSI, this is the 2D. The blue line is the half D, and the green line is the 1. There's a 1.5 and 2. So clearly the 2 was always better. The 1 and the 0.5 were similar until the highest pressure, where the 0.5 showed itself to be better. So then we go back and compare our predictions from the simulation with the experimental results. On the left is the predictions of the pressure for the 2D in red, the 1D in green, and the 0.5 in blue, compared with the experimental values. 2D, which was predicted by the simulation to have the lowest core pressure, indeed was able to remove more than the rest. The 0.5, which the simulations predicted would be marginally better than the 1, indeed showed itself to be better than the 1 size. So in summary, the effects of the swirl chamber size on the performance of the DYNASWIRL phase separator were studied. The velocity fields obtained by numerical simulation was validated by PIV measurements. The gas extraction performance did improve when the swirl chamber diameter was increased. When it was decreased, it did provide a marginal increase, especially at higher pressures. The larger size chamber resulted in lower pressure at the chamber, which implied a greater volume of air could be added. So we were getting 100% removal at very high void fractions, as high as 60%. So this is some of the artwork that's out in the hallway, but this is one of our simulations showing a bubble trapping in the core. The red bubbles are the larger, the green are middle, and the blue are the smallest. Thank you.
We have time for a couple of questions.
So, excuse me, very nice talk, and thanks for sharing some of the data and the results. The range in which this is applicable is the question, because for ecosystems, you're going to have very slow flow rates, or low liquid rates, high gas rates. There's other ones that are the reverse, where there's a lot of gas, hardly any liquid. There's all kinds of nuances when you get into a brine solution for urine processing, a separator for a water recovery system. So it isn't like one size fits all for these separator concepts. So could you give us the regime and where you're targeting here, and perhaps any thoughts about future concepts that you might be thinking about?
Well, we did all the work in water, so it was primarily for a water-based system. Now, we originally started for very low bubble concentrations, or air void concentrations, 10 to the -5, and then have gone since to much higher, 10s, 20%. Now, it all depends on buoyancy, coalescence as far as the capture. And the smaller the bubbles, the faster that flow rate has to be to induce capture. Does that answer your question?
Yeah, that's the right idea. Eventually, I'd just like to, maybe we'll talk later a little bit about, there's so much potential for this is really where I'm going. So maybe we talk later. Thank you.
Okay.
Okay, thank you very much. All right, next up is Dr. Howard Levine, who's going to bring plants back into the equation. Yes? If there's an issue. Okay, we're going to leave Howard for later, since we already have Steve Thorne is a research scientist for the Copernican project, and they have already loaded up his project, so we're going to go with Dr. Thorne. All right, thank you very much.
We on? Yeah, I wanted to thank, of course, Kevin and Annalisa for giving me this chance to talk, and I apologize if we've shuffled your timeline. And I'm not a doctor, but I appreciate the compliment. We're sort of, I'm from a motley group of Berkeley alumni members with backgrounds in physics and astronomy, and we are new to the field of space sciences and biology. But we take a more theoretical approach to things. And our real interest is in how inertial strain cycles get associated with moving reference frames. And so here we have two typical reference frames. And on the left, we have what we might call a Ptolemaic reference frame. And it's the frame that you see when you look around you, and it's very static. It's homogeneous and isotropic, at least on average timelines. But on the right, Copernicus taught us in the 16th century that the Earth is moving. And with that motion, of course, it's filled with rotations and oscillations. And they're small, of course. The rotation of the Earth is 10 to the -5 radians per second. And of course, we have this orbital motion that's factor about 10 to the -7. But what's interesting is when we contemplate trying to transition from one inertial frame to another, because of course that's what we do every time we move, when we're trying to move from the Earth into the ISS. And so my favorite scene of transitioning between inertial states is this 2001 scene, because it represents how not only do you have to catch the orbiting satellite, but then you have to adopt your rotation to match the satellite. And but it's not only your large scale rotation, it's all those inner fluids that also have to ultimately transition to that new inertial state. So that's our interest. And so it brings up a question. And if Copernicus taught us that the Earth is in motion, and Newton gave us the equations that say that these inertial strains are embedded in all scales, then why aren't they in our biological model? And I say that because we're sort of outsiders and new to the field. But what we find is we come to a conference here where everybody's talking about gravity and plants and roots and the smallest little deviation in G. But we were at a conference in San Francisco a year and a half ago, the Biophysical Society, and there were 7,600 people there, and the word gravity wasn't mentioned once. And you can open up textbooks, as now we have had to do as we try to bring up our biological side. Cell is an example, it's this big. Other than a couple throwaway lines, you won't find the word gravity in there. And so the question is, maybe, does the motion of the Earth matter? And it's a funny question, of course, but we're moving at 30,000 meters per second. We're rotating at about 465 meters per second. And actually our orbital velocity is Mach 88. And does that matter? We don't know, but we're interested in finding out. Now, if we ask NASA, NASA will say it matters. That's why NASA launches to the east, because there's a small energy advantage that's gained when you launch in the direction that the Earth is rotating. But you only understand that when you step back to this Copernican frame of the sun and calculate your energies from that domain. And so it's also how NASA explains the gravitational slingshot effect, when they boost the speed of a satellite by throwing it into the gravitational well. You have to model these things in the reference frame of the sun. So maybe a more appropriate question is, does the motion of the Earth matter to life? And that's a much more complicated question. And I told David Tomko that I wouldn't fill this with equations, but here's just a schematic of the players involved in this game. And of course, we have the Earth, the Moon, the Sun, and we have a Berry center that governs our orbit. And then we've got, of course, the little man and these gravitational vectors. There's also centrifugal vectors that have to be included in this. So it gets very complicated, and I don't want to start with this. Instead, let's go to something more simple. And this is the Newtonian model where we treat things as a point. And if you're an organism traveling on the Earth, this is a very boring life. There's just a smooth, static condition, almost like when you look in your coffee cup, okay? But that's not the reality. The reality of our true path of earthbound organisms is much more like this yellow course. And because as we're orbiting at 30,000, we're also wobbling due to the speed. We're rotating with the Earth. And those induce these small-scale internal strain cycles. But what's also interesting is that in the reference frame of the sun, if you calculate the speed at which a body is moving, between noon and midnight, we have to accelerate almost 1,000 meters per second, 4 times as fast as an airplane travels. And that is because we've got 30,000 meters per second. This way is an orbit, and we're rotating, and at noon, we're actually rotating opposite to our direction. We subtract the 465. So fundamentally, what every organism is being subjected to on Earth is this acceleration of 1,000 meters per second over 12 hours, and a deceleration, and an acceleration, and a deceleration. And it's very precise. And then on top of that, we've got this moon friend, and this moon is orbiting around us, and that kicks us. It kicks us into this wobble. So imagine being in this car, and you're driving down stoplights that are slowing and speeding up, but you're also zigzagging. So a more realistic reference comparison is this is riding on the teacups at Disneyland, and that's really our inertial state. That has become our home. So all these molecular partnerships and all these proteins and these metabolic pathways have been tuned to this inertial strain cycle because it's embedded in our system for 3 billion years. So not only are we on the teacups, but a more realistic picture is to put those teacups onto whatever, we've got a roller coaster, because that's more representative of the path. Now, does that matter? If you're a rock, it doesn't matter, because when you accelerate a rock, the whole system accelerates together. There's almost, the internal stresses dissipate so quickly that there isn't internal movement. But if you're a bound fluid like a cell, and anything that's less viscous than the mean, or more viscous than the mean, and when you've got all these different viscosities, you accelerate the bound fluid, the things that are more dense stay behind, and the things that are less dense are actually pushed forward. And that induces a small little torque into the system. And that torque can be, if you couple those motions, you can use that torque for local work or timing uses. And so now we're not going to look to try to calculate what these forces are, but here's a fascinating study we did. And we consider these just coincidences at this point. But we said that if we're not going to calculate these small scale detail, maybe they'll show up in the large scale energy patterns of our biology. So we calculated how much energy would it take for an ATP molecule to lift it in the reference frame of the sun. And it's a simple question. Normally the energy is mgh when it's relative to Earth. And so we just take G being the sun and H being then the diameter of the Earth. And it came out to be 36 kilojoules per mole. Now our biologist friends said that's a cool number because that's also the energy of hydrolysis, roughly so. And we said, okay, that's a coincidence. And then we looked at how much energy it would take to lift a human body from one side of the Earth to the other, because this is what happens between noon and midnight. Between a series of gravitational pulls and sort of viscoelastic shoves, we get pushed further from the sun in that gravitational field. It turns out that the energy that is required to lift my body is roughly about 1,130 calories. And those that work in physiology might recognize that as being roughly your basal metabolic rate. That's the minimum amount of energy that's needed to sustain my system over the course of one day. So these two figures that, again, we just consider as coincident at this point, but they're kind of cool coincidences because they sort of connect an inertial property, possibly, to the unit of energy released in our biology and the large-scale system-wide energy. We don't pretend to understand it, but that's actually one of the reasons why we're here and in other conferences. So what does that give us? If you look at the strain cycles inherent to Earth's orbital motion, first, of course, you're going to notice the circadian periodicity. As I mentioned, there's this 24-hour velocity increase and decrease, all right? And then we've got a circulunar periodicity. And that's actually been detected by a couple, I was there a month ago in Göttingen. Some Max Planck researchers have detected an inertial strain cycle correlated to a lunar periodicity. And they've also, I'll stop there, but that's governed by our motion around the barycenter. We have a seasonal periodicity, of course, because our orbit is elliptical. And we're speeding up for 6 months and we're slowing down. It's that same car scenario. And that actually has a 7% influence on our energy numbers. And if you look at our pattern, if you look at our actual motion of a body as it's moving like this and twisting in the reference frame of the sun, there's a little torsion. There's a chiral strain cycle. And it gets wound at this inertial level into all systems. And our biologist friends said again, well, hey, amino acids have this preference for a certain chirality, and it's not explained. We're not saying that this is the explanation, but again, we just find this sort of correlation. All we're really doing is pointing out nothing's new here. Everything that we're pointing out is physicists and astronomers have said there's all these patterns inherent to our orbital motion, and and biologists have say, hey, all these similar patterns are in our physiology. So we're just saying, hey, maybe these two sides need to come together a little bit, but that it's just fascinating. And the other thing that's fascinating is that all of these cycles, orbital strain motions, are 1,000,000 times more precise than a Rolex watch. Now, that may ruin my funding opportunity with Rolex, but it's the truth. You can't build a biochemical chain reaction that has near the precision of these orbital strain properties. And if you look at these numbers, that what we're talking about is trying to find coupling mechanisms to these finer scale strains, which, like Chris Wolverton's group the other day, showed that gravity sensing roots were at least picking up gravitational strains of 003. And we're just looking to find, point out that these strain cycles exist at even these finer scales, and maybe things have coupled to them. I won't go through these fascinating experiments. We're trying to be, on our site, the copernicanproject.org, we're trying to post experiments that might support these notions. They're not by us, they're by other researchers around the world. And then I'll just close by saying that the issue as we move into space is that if our system has coupled to these strain cycles, okay, that for 3 billion years, this geometry evolution of proteins and metabolic pathways, if that has become our home, and then we move to Mars, or if we head towards Mars, Mars actually has that similar periodicity as the Earth, but it's that transition in between where there's 7 months and we don't have those inertial cues. And maybe that's a problem we need to consider, and there's ways to test for it. But I'll close there and let the next continue. All right.
Thank you very much. I think we'll have to forego questions until after the break, but that was fun. Thanks. All right, next up, Howard Levine. I hope. Howard Levine is the chief scientist down at Kennedy Space Center and has done a lot of work on organizing how we grow plants in space with some of the constraints that we've heard about this morning. Thank you, Howard.
Thanks. Can you hear me? Good. That's not my presentation. Good. Okay, so we set out to design a new way of growing plants in the veggie system on the International Space Station. So, and the key aspect of it is it's a passive nutrient delivery system. So we call it the Passive Orbital Nutrient Delivery System, or PONS. You know, veggie has been a big success the last few years. There's all different kinds of plants. There's a lot of knowledge that we've gained in terms of crop production in space. But having said that, there are some issues with the current baseline nutrient delivery system, which essentially, it has this reservoir that is used to transfer water to these 6 independent, we call them pillows. And sometimes that works and sometimes it doesn't. There's also issues at the pillow level in terms of sometimes the plumbing could get clogged up. It's hard to tell how much water is remaining within, so it's a kind of a very labor-intensive activity to try and monitor when to add water, when to not. And the crew has to independently add water to these 6 pillows. And as a result, there's cases where there's sporadic germination in some instances. There's been cases where there's been overwatering, and when there's overwatering, you can sometimes get opportunistic pathogens that colonize your biology. So forgive me, my voice is heading out. Don't ask me what this is. We set upon a little project at Kennedy to develop this alternative approach. And essentially what you have is the plant cylinder that contains the substrate. You have an external reservoir that can contain up to 500 milliliters of water, and they go together. So it's kind of a hybrid system. And again, the initial constraint was no moving parts, no electricity, so entirely passive. Here's some of the details in terms of how you can fill it. Oh, look at that, I'm out of power. Okay, so, but you kind of follow that. Let's see. We use this Oasis root cube foam to keep the substrate from floating out of the plant cylinder. But it's also very important because it's very hydrophilic, so that when there's excess water up here by the capillary mat into which the seeds are planted, it can wick the seeds, wick the water away, which is very important. So this was our first prototype, just to give you an idea. There are some oxygen permeable not going back. There it is. Oxygen permeable areas to try and bring oxygen in. There's also oxygen that comes in from the top through the plant cylinder. There's a root cap to maintain high humidity during the germination phase. And this was our first experiment with lettuce. We had 3 lettuce seeds planted into within this capillary mat. All 3 germinated. You can kind of follow leaf 1, leaf 2, leaf 4. And we did these experiments under conditions similar to what the veggie experience is on ISS. We took this experiment through to the point where we could do a harvest. We harvested about 35.6 grams, which you might consider to be about a salad for 1 crew member. And under this cut and come again approach, you can see in the lower left what it looked like immediately after harvest, 4 days after harvest, 12 days after harvest. And then we did another harvest, our second harvest. We got about 28.8 grams, maybe again another bit of a salad for 1 crew member or shared. The interesting thing is, though, that the leaves then started to undergo a morphological, physiological change. They became more velvety, and we started to get the beginning of a stalk, which then, you know, and I had never worked with lettuce before, so I found this amazing. You know, the stalk actually grew up and out, and we could follow that through. We got flower formation, the seed formation, so it was a self-pollinating situation. 95% of the seeds germinated, so they were self-compatible. We went on, we did our second prototype design. We kind of got a little more sophisticated with the oxygen input ports, you know, make them replaceable. So since we're NASA, we call this orbital replacement units. We put in a little window on the side so we could monitor the water level over time, and we collected some data on that. So with that second prototype, we did 2 parallel experiments, and these plants are mimicking what was being grown in veggie on ISS, so that's why they were chosen. But on the left, you can see Chinese cabbage. Again, 100% germination. We thinned them out. You can see the removed seedlings on the right is tomato. Again, 100% germination. And I want to draw your attention in the lower right, the foam. You can see that the foam is wetted in the immediate vicinity of the capillary mat. So that's just demonstrating that when you have excess water up there in the immediate vicinity of the seed, you can wick it away, and that's very important. So when we harvested the Chinese cabbage, it kind of looked like this. And I guess the main thing I want to point out here is that the roots grow out from the plant cylinder into the external reservoir. They grow out through the capillary mat. And there was capillary mat bringing water in from the bottom, but also from the sides. So you see here that they can penetrate. So even without any extra external slits or whatever, the roots eventually have access to the water in the external reservoir. These are the results from the ponds experiment. Flowers started to open at day after planting 34. In the bottom, day after planting 48, I just want to draw your attention to the foam is compressible. So when the stem begins to really expand, it allows that to happen. There were other materials that were tried that cut into the stem. So day after planting 86, we have fruit. Again, good flower development. We harvested those tomatoes. We did a second harvest. We harvested those. We did a third harvest. You know, the plant is generally going downhill, but you can keep it going. We developed a method where we supplemented the nutrients in the reservoir with commercially available Miracle-Gro, a dilution of it. Initially, all of the nutrients come from the slow dissolving fertilizer pellets, as similar to what Joy Amasa and others have used in the arsenite. So here's just some data, and again, okay. So I want to point your attention here. So we have data, cumulative water usage over time, and you can see that it actually reached in excess of 20 liters over the course of the experiment. And then the red boxes are daily water usage rates. So you can see how that's much larger at the beginning, and it especially drops down when we do our harvests, and eventually the plants peter out. Here was our third prototype. We added concave corners to kind of prevent the edge creep of water and microgravity up the corners. We enlarged the oxygen diffusion ports, put them on all four sides, some on the top, because we realized that oxygen availability is one of the critical issues in this kind of a passive system. So here are the parts. This was our fourth unit. We finally moved over to black PVC and plexiglass, so to prevent light penetration without just wrapping black tape around it. So we grew zinnia. We actually had zinnia going almost for a full year using this external supplement approach with Miracle-Gro. Because here we are at this conference, we know a lot of folks are interested in Arabidopsis, so we grew Arabidopsis. Again, we had good germination, a nice rosette formation, all the way through, full CSIC formation. We did a larger scale ponds unit that actually had 6 ponds units that would share a common reservoir. So this is kind of a top view. Again, 100% germination in all cases, a nice crop. We had some extra parts, so Larry Koss, one of my co-authors, built a tubular design. So all of this, is testing and prototype development. We subsequently became involved with, I pitched it to Tupperware. And Tupperware was very excited about doing something in space. And so we entered into a partnership with Tupperware and TechShot, kind of to guide them through the flight process. And so you can see the flight design is there in the upper left. And I have a unit here, and it's available at the TechShot table. And you can see it's just like the 6 pillows, there'll be 6 ponds units. And there's just a ground study with some, looks like Mizuna, and a little bit of a blow up. But I hope to tell you about some of the successful flight validation experiments at next year's conference. But right now, That's pretty much where we are. Any questions?
All right, one quick question if we have it, otherwise we'll move on to our last talk. All right, catch Howard in the break. Thanks again. All right, we're going to finish up the session with Tyler Hatch, who comes from NASA Glenn, and he's going to put together all the stuff we've talked about.
All right, it's great to be here. I'm very fortunate we had some fluid talks leading up to this, and then the plant talks also. So this does kind of tie everything together. So my name is Tyler Hatch. I work up at NASA Glenn in Cleveland, Ohio, and this is our plant water management experiment. Okay, so I'm going to go over who we have on the team for PWM. We'll look at some of the science background behind it. I'll take a look at what we have for the experimental hardware, and then we'll get into the analysis of the system and then our future plans for it. So Mark Weisslogel is our PI for the project. He's been a lot of fun to work with, and he's done a lot of work with the similar plant experiments too, so we're great to have him on the team. John McQuillan is our fluid physics discipline lead up at NASA Glenn. I'm project scientist for it. We have Kelly Bailey as our project manager, and then Zin has been doing the engineering and building the hardware for us. Okay, so as many of you may know, we have a lot of challenges that we face when we're trying to grow plants in microgravity. Those come from aeration. You have different gas and fluid interactions. There's diffusion of nutrients, whether you have a slow release pellets or some type of liquid. And then the root growth, that can change at any point in time when you're in the germination stages, and then even later on in the kind of the harvest stages of the plant. You have very different characteristics you have to look at. And so far they've seen a lot of water migration and different issues that come with that, at least in some of the veggie experiments. And we'd like to see if we can kind of look at those and look for what are some simple, robust methods we can use moving forward for advanced concepts for some of the larger scale stuff. Okay, and then I'm a fluids guy, so I thought I'd throw this in. Mark Weisslogel tries to drive this home every year, so I thought it was important to put in, but at least on the ISS, virtually every major system on there has some type of fluid interactions that you have to deal with. And those can either work to your advantage or your disadvantage, depending on how you look at it. And these go from some of the fundamental boiling condensation research to applied research in plant growth, for example, and even some of the exploration where you have cryogenic management you have to think about. Okay, so for plant water management, these are the main goals we're trying to look at. Specifically, we're really focusing on the root zone and how we can look at hydration and aeration in that area. I'm not going to read through all these, but the big takeaway is that all these different bullets start with demonstrate. So PWM is a technology demonstration where we're using a lot of the techniques that Mark has developed with his capillary flow research, and we're applying that to some of the plant growth systems we have now. Okay, so PWM is broken down into 6 major concepts. We have the first 2, which are paired up. The first one is a soil-based concept, and then we have a hydroponics demonstration we're working on. And then the next 4 of them look at some of the more advanced techniques we're trying to capture with the root accommodation zones. We have a larger area for the plants to grow. And we look at some new techniques for aeration and properly hydrating the root zone. These are all pretty simple. We'll send them up to the ISS, and then they're all operated on the MWA, which is a mobile table workstation. It's all crew-tended. It's task listable, so they can do a few of them fairly easily if we get a couple hours for crew time. And operationally, it's pretty simple. We'll just have a video set up to take video and track the flow rate, and then we can send that down and do some analysis here on the ground. And the picture is one of Mark Weisslogel's other experiments, the capillary structures experiment, and this was kind of based off of that technology. Okay, so this is a look at the hardware we've got. These are the 2 ground units, and we have models actually on the ISS right now for the first 2 out of our 6 concepts. Number 1 is our soil concept, where we have a 3D printed test stand, 3D printed test cell, and then the reservoir standpipe that we use with our test fluid. the soil we're using is arsenite, which they've been using for years with veggie, and all the tubing and the syringes are all COTS off the shelf, just to simplify everything. And just a disclaimer, there's no real plants involved with this. We're using different types of sponges, felts, and foams as our stand-in, and those are acting as our simulated plants for this experiment. And it gives us a lot of flexibility to design different examples for the roots that we can demonstrate up there, whether you have a tap root or some of the more fibrous roots. We can just take one out and drop a new one in, and it's very simple to test out a lot of different concepts without having to look into a lot of the biology that goes into it. And then the hydroponics unit, same idea, it's all 3D printed, off the shelf wherever possible, and then we have a wedge test cell that's based on a lot of Mark's work. There's a sharp interior corner there, so we can use it as a capillary system to drive the water through in case the pump we have fails or there's some blockage issue that comes up. Okay, and this is a schematic of the flow loops. So for the soil one, we have a syringe to prime the system, and then we'll use that with our test fluid. Once it gets to the standpipe, we'll take out the syringe and let the simulated plant draw as much as it needs. It's more of a long duration test, so we can look at it over 30 minutes an hour and really track that evaporation rate, which would really inform a lot of the fluid designs that we might need on the ISS. Hydroponics, we have 2 syringes, mainly for priming, but it's pump driven, and then that'll be crew tended, so we can have the astronaut adjust all the flow rates and map out what are the stability regimes for this type of hydroponic system. Okay, here's a deeper look at some of the hardware for the soil experiment. As I said, it's all COTS off the shelf wherever possible. We 3D printed the test stand and some of the inner components. Our test fluid is actually from the crew pantry. We're using a red drink fluid as our stand-in for some of the nutrient solutions you might see in a plant growth system. And the simulated plant, we're using a rayon felt chamois as our simulated stand-in for what you might have in a real system. Very similar for hydroponics, all 3D printed or off the shelf. And the actual test cell we have to hold the simulated plants, we can hold up to 3 of them in there at a time and do some testing with that. And then it has a removable lid, so we can test some of the stability and look at how the system will behave with an open system, just using capillary as our containment for that. Okay, so the test fluid, we are using a mixture of the tropical punch they have in the crew pantry. So it's really great from a fluids point of view. We can get some really good visualization and contrast on there. And the actual soil media we're using is a mixture of this arsenite, which they've been using for years, and we have magic sand, which is a non-wetting aeration kind of media we're using to see if that can help address some of the aeration issues they've been having with plants over the years. And these are the plant stimulants we have. As I mentioned, it's the rayon felt, and we have a mixture of nylon and different strings in there to help with simulating the roots. And we can use different types of designs and geometry to simulate, for example, a plant in the germination stage where we have a throttled uptake. And that gives us a lower flow rate going into the plant, a lower wicking rate, so that we can look at basically the whole life cycle of a plant in this kind of simple experiment. Okay, and then operationally, it is fairly simple. We'll have the crew take out the kit, they'll assemble it, and then set up the video camera for recording. It's all right there on the MWA, so it's right in the middle of the cabin. It's open to the cabin air. You don't have a lot of containment to worry about. And they'll start the system, they'll prime it, turn on the pump, and then we'll work with them to figure out kind of what the regimes are and the flow rates we can handle in these type of systems. The soil one is great because they can leave it alone and give it some overnight evaporation, so we can have a really long duration study to figure out what the evaporation rate would be for this type of system. And then after that, they'll pack everything up and then stow it or toss some of the plant simulants after they've been used. And that's it. It's very quick. It's task listable. We can do a lot of great science pretty quickly with this. And this was actually an experiment we did from the initial concept to sending up the flight hardware in about a year. So it was very fast paced. And it's really important that we do this on the space station because, at least for the soil experiment, we have a 30-minute to an hour demonstration that we're doing. So we can track the evaporation rate over this long period in microgravity and then compare that to a 1G test and see what the difference is. Because it's crew tended, we have more potential to test out the stability, whether it's from G-jitter or perturbations as they occur. And we can look at different bubble responses and see, kind of fine tune it with the crew working right there with us on it. And as I said, we did this whole experiment in about a year. It's very similar to what Mark has done before with his capillary flow experiment, and then the capillary structure is a new one he's working on. So we used a lot of the same design principles and the similar styles of hardware that he'd used, where capillary structures had a lot of 3D printed and COTS off-the-shelf work. We tried to replicate that as much as we could to see if we can get something up there pretty fast. Okay, so this is a look at one of the soil experiments where we have our a simulated plant in the 3D printed test cell, and then we have a mixture of the non-wetting magic sand soil, which is blue in there, and then it's with the arsenite. And we're thinking you can use different types of geometric patterns in there to really control your uptake and limit what the plant can see while giving it room for aeration as it's growing. And this is really cool because we have the arsenite and the magic sand are really about the same porosity. We were able to sieve them down to the same size, and we can really track the moving, the contact line as it moves up through there with our test fluid. I'm still working on some of the analysis for that, but moving forward, we think the hydroponics is really the way to go with a lot of this stuff. So it's based on Mark's CFE work, where we have a sharp interior corner to pin the test fluid there. And then using capillary forces, we can have a continuous draw. Even if the pump fails or encounters some type of blockage, it'll continue to work and keep the plant hydrated, the simulated plant. Okay, and then based on this, we can look at our free surface profile and find out the flow rate from that. It's all based on the videos that we'll downlink here to Earth. And we can map out the stability regime based on what flow rate we have, how much liquid is in the system. And this is about a 6-inch size, just for reference. Okay, and so far we've looked at 4 main regimes, at least on the 1G demo of everything. You have stable flow, where you have a continuous liquid stream the whole time. If you have a higher flow rate, then you can start to get ingestion, where you pull in air bubbles into the outlet of the system, and those can go all the way through and come back on the other end. If you have a higher free surface profile, then if we increase the pump rate, then you'll start to actually lose some of your fluid, and we'll have embolism, where it bubbles up and then it'll eventually eject out some of the liquid. Okay, so here's a video. This is a 1G test version that Mark made. It's about a 1/3 scale of the flight hardware. Okay. Let's see. Okay, so we're running it, and then we drop that lower left end, and you can start to see it pull in air bubbles, which come back around. And then we think that might be a good way to help some of the aeration issues we're facing with plants. This is just a demonstration of it, but we're looking at some of the more advanced concepts at how we can control this air process to look at the aeration with it. Okay, here's embolism and injection. So if we have a higher profile, then the high flow rate will mean we start to lose some of our water over time. So that kind of maps out where our stability limits are for this type of system. Okay, and as I said, we're still working on what these operational limits are, but kind of the long-term goal is we'd like to see maybe a hydroponic system like this on one of the up-and-coming systems for Gateway, or even some of the Martian systems. Okay, and this has been really great to show that you can use capillary flows as a method of containment without a lot of external containment or containers. It's open to the cabin air for this experiment, so that's great for us. We're using this fruit punch drink as our stand-in fluid, which is closer to a lot of the poorly wetting fluids we have in most of the real systems up there. And then it's great because you can do 3rd scale demos for proof of concept and testing on the ground, which here's another example of one of those. Okay, so right now we have the first 2 units up on the ISS, the soil and the hydroponics. We're waiting for crew time to operate those, and we've got a couple more in the pipeline that deal with the advanced concepts for parallel hydroponic trays and the root accommodation. Those should be launching next year, and then big shout out to all the KSC folks who've been helping us kind of map out the simulated plants and what we need for that. Joya, Oscar, Ralph, Tom and everyone else. So thank you. I'll be happy to take any questions.
All right. Well, thank you very much. And any additional questions, if we could please maybe have them at the break, because we are out of time. Thank you very much.
Thank you.
Right. So that closes our session for the morning. Remember that the high school poster session is running right now and will be running kind of in the background as well all morning. And thank you very much, everyone, for your patience and attention as we work through the morning. Thank you much. One more for the speakers.